Hello, everyone. My name is Matthew Cooper, and today I'll be giving a presentation providing an introduction to Mendelian genetics. This presentation is geared towards AP biology students, although it could be applied to AP anatomy and physiology as well. This presentation is for Biology 510, a comprehensive overview of cell and molecular biology through Grand Canyon University with Dr. Sean Stacy. So the objectives for this presentation today is first to discuss Gregor Mendel and his history, so who Gregor Mendel was. Secondly, we will describe Mendel's pea plant experiments and the results of those experiments and their statistics. We will then explain Mendel's 3 laws of inheritance and how they apply to all genetics, or the majority of genetics at least. And then finally, we will demonstrate the use of Punnett squares and pedigrees to track inheritance as well as predict potential outcomes. So, who was Gregor Mendel? Gregor Mendel was born on July 20th, 1822 in Hazendorf, Silesia, Austrian Empire, which would be modern-day Czech Republic. He began his academic career studying mathematics and physics, where he did have some success. Unfortunately, though, his family was not very wealthy and couldn't afford to continue his education. But luckily, his professor directed him towards the Augustinian order at St. Thomas's Abbey, which he joined in 1843. This order of monks and priests held education very highly. And actually ran some schools. Eventually, in 1847, he was ordained a priest. And when he was working in this abbey with the Augustinian order, he transitioned from physics and mathematics towards botany, although he did still utilize statistics. Now, through his study of botany, he was given the opportunity or the task to study hybridization and the transmission of traits to future generations by his abbot in 1854. Now, his findings with these experiments and all of this information led to the moniker Father of Genetics, which to me is somewhat funny due to the fact that he is also a priest, so we would call him Father in that regard as well. Now, before we move into the actual science, I would like to cover a few important terms that you'll hear me say, that you'll see in the PowerPoint and things like that, starting with allele. Now, an allele simply means a different version of the same gene. Okay. Now, we can refer to alleles as either dominant, which is a trait or allele that only needs one copy to be expressed, or recessive, which means a trait that or allele that needs 2 copies to be expressed. Now, after we get those alleles organized and figured out, we can also discuss things called phenotype. Okay, the phenotype would be the physical appearance of an organism, and the way I like to remember that is that physical and phenotype both start with a PH. And then we can also discuss genotype, which is the actual genetic makeup of an organism. And that one for me is easy to remember, but if not, just remember that gene and geno, it's basically the same word. Now, when we're talking about genotype, we can discuss it in multiple different ways. The first way being heterozygous. Heterozygous means that an organism has 2 different alleles for a trait. So it would have one dominant and one recessive allele combined. And then the other way to discuss it would be homozygous, which basically means that an organism has 2 of the same alleles for a trait. So it could either be homozygous dominant, which means it has 2 dominant alleles, or it could be homozygous recessive, meaning that it has 2 recessive alleles. So let's move on to Mendel's experiments. And you'll see here I have a pea plant, and that is for a very important reason. It is because Mendel chose to study the edible pea for 2 really important reasons. The first reason being that it is easy to grow and pollinate, and the second is because it has a very high rate of seed germination. Those 2 factors combined led to a large amount of offspring, meaning he could have a large sample size, which is very important in statistical analysis. So when he was pollinating and germinating these pea plants, he was very careful to control the pollination of pea plants. going as far as to utilizing a paintbrush to pollinate the plants, and also removing the stamen from the flower once it was pollinated, and also removing extra flowers that were unnecessary, to just make sure that he controlled everything to do with the pollination of these pea plants. So in a study, he chose to focus on 7 either-or traits, as I like to call them, in true breeding plants. Now, what true breeding means is that they are purebred. So if they had yellow peas, it was always yellow peas. It was homozygous dominant. And if they had green peas, it was homozygous recessive, so always producing green peas. And by either or, I mean that it was either this trait or that trait, either the dominant or the recessive. There was no in between, and only those 2 traits. So if we look here at this picture to the right, we can see that he focused on pea shape, which was either round as your dominant or wrinkled as your recessive. Pea color, which was yellow as dominant, green as recessive. Flower color, purple or white. Flower position, axial, which means along the sides and the bottom, or terminal, which means at the top. Pod shape, which was inflated or pinched, which means it was holding the actual pea itself either loosely or tightly. The pod color would be, once again, either green or yellow. And then the plant height, either tall or short. Now, for me, with pod color and pea color, it is odd that the dominant trait for pea color is yellow, but pod color is green, whereas it's the opposite for recessive. Now, during his experimentation, which took around 7 years, he kept very detailed notes of all the breeding outcomes across 3 generations over and over again for each trait. And those 3 generations would be the P generation, which is the original generation, the F1, which was the first set of offspring, and then the F2. Now, before we get into the experimental results, I would like to take a look at these 2 pictures here. So if we look at the first picture, we can see that he was focusing on one trait, so pea color. And then the genotype of the yellow pea was homozygous dominant for this parent, and then green pea would be homozygous recessive for that parent. So he cross-fertilized those 2 together and saw that he received all yellow peas. Later, it was determined that those would all be heterozygous. And he knew that because he had crossed a purebred yellow with a purebred green. Now, during that first generation, or the F1, he self-pollinated those yellow pea plants together, which led to the offspring that we see here below. Now, that led to 3 yellow pea plants to every 1 green pea plant. So it was a 3-to-1 ratio when he was looking at that monohybrid ratio for phenotype. And he found that was the case with every single trait pair that he observed. So not just pea color, but also pea shape, pod shape, things of that nature. So now whenever we look at a dihybrid, which means that we look at 2 traits at the same time, if we have a double homozygous dominant trait, such as the yellow round pea, and we crossbreed it with a double homozygous recessive pea, such as the green wrinkled one seen in this picture, and we cross-fertilize those, we notice that we will always get a yellow round pea. And that would be double heterozygous for both of those traits. And then if we self-pollinated those, or self-fertilized those, we would once again end up with a ratio of different colored and sized, or different colored and shaped peas. So the dihybrid ratio would be 9 to 3 to 3 to 1, meaning 9 yellow round peas, 3 yellow wrinkled peas, 3 green round peas, and only 1 double recessive or green wrinkled pea. So Later research found that genotypic ratio was often different than the phenotypic ratio in many cases. Some cases not so much, but in most cases we can see that ratio is different. So if we look at that same monohybrid cross that we looked at earlier, we can see that there is a monohybrid ratio or monohybrid genotypic ratio of 1 to 2 to 1. So 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. So all of these findings and all of this information that Mendel found led him to create 3 laws of inheritance. So the first law of inheritance was called the law of dominance. And basically this states that a dominant allele, such as yellow pea color in our example, will always express itself over a recessive allele, or green pea color in our example. So yellow pea color, or uppercase Y, because we always assign the uppercase letter to the dominant trait, is dominant over green pea color, or lowercase y. So if a plant offspring inherited that uppercase Y allele from one or both parents, it will have a yellow pea color. So whether it is homozygous dominant or heterozygous, it will always be yellow. Now, the second law of inheritance was known as the law of segregation. So basically this states that when chromosomes separate in meiosis, each gamete, which is egg or sperm, will receive only one chromosome from each pair. So for example, if a man has alleles for brown hair and blonde hair, then he is heterozygous, and we'll say uppercase B, lowercase b for hair color. His sperm cells can obtain the allele for brown hair, uppercase B, or the allele for blonde hair, lowercase b. So each sperm only gets one of those alleles. Okay, so that's very important to acknowledge that if you receive 2 chromosomes for that, it can cause some problems. So Mendel's third law is the law of independent assortment. And this states that the assortment of chromosomes for one trait does not affect the assortment of chromosomes for another trait. This allows for any combination of maternal and paternal chromosomes to be passed on because homologous chromosomes line up randomly during metaphase. So basically that could mean that the recessive allele ends up on the left-hand side instead of the right-hand side, or vice versa. And that can create combinations that are innumerable. So, moving on to things that we do now to calculate and to predict genetic outcomes. So pictured here is a Punnett square, and this is a visual representation of potential genotype and phenotype offspring. And we saw one of these earlier in the discussion of Mendel's results. Here it's talking about flower color, obviously. We have 2 heterozygous plants here that are both purple, and we see that we receive that 3 to 1 phenotypic ratio of purple to white flowers, and we also receive that 1 to 2 to 1 genotypic ratio of 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive offspring. Now, we need to know how to utilize a Punnett square, often because it shows up on your AP exam. So, the first step you want to do is determine your parental genotypes. For this example, I'll be providing that to you. We will go over another example of how to do it on your own. So in this example, we'll have double heterozygous, so uppercase T, lowercase t for both parents, uppercase T standing for tall and lowercase t standing for short. So the second step you want to do is you want to draw a 2x2 grid. Should look a little something like this. And with that grid, you want to place your first parent's alleles above the boxes. So it would be 1 letter per box as shown there. Then you would want to take parent 2's alleles, and put them to the left of the boxes in the same manner, 1 letter per box. Now, The third thing you want to do is to fill in offspring genotypes by combining the parental alleles inside the grid, where you would bring your big T's down and across, and your little t's down and across. Okay, so I like to put my big or uppercase T's first whenever I'm doing these letters, but it doesn't really matter as long as you understand what heterozygous and homozygous means. So the first square would have two uppercase T's. The right-hand upper square would have uppercase, lowercase, and then we have lowercase, lowercase. Now, the final step is to analyze your results. You can infer the phenotypes from derived genotypes. So we can observe and infer that we have 3 dominant phenotypes and 1 recessive phenotype. And then we can calculate those genotypic ratios and phenotypic ratios. So our genotypic ratio in this case, once again, would be 1 to 2 to 1, because we have this 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. And then our phenotypic ratio would be 3 dominant and 1 recessive. So let's look at another example that requires you to answer some potentially more complicated questions, because these type of questions will also arise on your exam. Excuse me, I'm sorry. So in this example, we'll say that in humans, tongue rolling is dominant to non-tongue rolling. A man who is homozygous for tongue rolling is crossed with a woman who is heterozygous for tongue rolling as well. So you will be tasked to create a Punnett square and answer the following questions. So our first step, once again, is to determine the parental genotypes. So it says a man is homozygous for tongue rolling, and we see that tongue rolling is dominant. So that means that it is going to be an uppercase letter. And then the woman is heterozygous, meaning she has 1 uppercase letter and 1 lowercase letter, or 1 dominant gene or allele and 1 recessive allele. So that shows us that the parent 1 is uppercase R, and parent 2 is uppercase R, lowercase r. Then we want to draw our box and put in our parental alleles above and to the left of those boxes. So like that. Okay. Now, the next thing we need to do is we need to create our genetic offspring, or predict our genetic offspring. So that first square would be big R, little r. We have big R, little r, double big R, and double big R. So that gives us a genotypic ratio of 1 to 1. And that is very important that you do not put 2 to 2. Your AP readers will count that incorrect because ratios must be simplified. Okay? And the phenotypic ratio would be 1 to 0, or all dominant, all tongue rolling. So the easiest way to remember to simplify your answers, or how to simplify your answers, is for every 1 heterozygous, we have 1 homozygous. And for every 1 dominant phenotype, we have 0 recessive phenotypes. So let's look at our following questions. Okay, so what is the percent chance that these 2 people will have a child who cannot roll their tongue? So if we look at our Punnett square and our phenotypic ratio, we see that they always have the dominant trait, meaning that 0% would be the chance. So it is a 0% chance that they would have a child that cannot roll their tongue, unless, of course, there is a mutation, which we'll be discussing later on. So the second question says, what percent of the children are homozygous dominant? So if we look for homozygous dominant, we can look at our genotypic ratio and see that we have a 1 to 1, which is heterozygous to homozygous dominant. So that would make us have a 50%, or that number would be 50% of the children. So our last question here says, what percent of children would show the dominant phenotype? And as we can see, just like our question for question number 1, or our answer for question number 1, it would be the opposite. The phenotypic ratio is 1 to 0, meaning we have a 100% chance of the dominant phenotype being shown. Okay, and the final thing we're going to discuss is how to read and utilize pedigrees. Pedigrees are used when attempting to track genetics through several familial generations, and they provide a large amount of information utilizing symbols, as we can see here to the right. Roman numerals denote generations, squares are males, circles are females. Colored in means that you're affected, not colored in means you're not affected by the trait being tracked. Horizontal lines are marriage, and vertical lines connect offspring, and the order of offspring is denoted by Arabic numbers, as well as moving from left to right, similar to how you would read a book. Now, there are ways we can use pedigrees. You can use them to determine the type of trait being tracked if it's not given to you. And what I mean by type of trait would be autosomal dominant, meaning it's on your autosomal chromosomes, um, or your autosomes, autosomal recessive, X-linked recessive, which means it's on your sex, the X of your sex-linked or your sex genes. Um, and there are some steps to use when trying to determine this. So the first step would be if there are way more males than females affected, or if there's a noticeable number of colored in squares above colored in circles, then you are more than likely looking at X-linked recessive trait, and it is safe to generally make that assumption. Step two, you would go to if that is not the case. So what you would want to do is look for two parents that are the same. Okay, so in this case, we have two parents that are the same, and have a child who is different. So that child right there is different from the parents right there. So you would label the child homozygous recessive, for example, double lowercase r, and the parents would then be labeled as heterozygous. So to further move into that, to determine whether it's autosomal recessive or autosomal dominant, you would look at the shading of the different people or organisms that are being looked at. So if the child was shaded and the parents were not, then the pedigree would always be tracing an autosomal recessive trait. And if the parents were shaded and the child was not, the pedigree would be tracing an autosomal dominant trait. So let's apply these steps to this pedigree here. So for step one, like it says, check to see if more males are affected than females. So we count our affected individuals. There are 3 females and then 5 males. So that in and of itself stops us there and lets us know that we are looking at X-linked recessive traits here. Okay, so if you need to review this information, I've provided you with 3 links to some really good videos. You have AP Bio Penguin Insta Review, which reviews all of AP Unit 5 on heredity, including material we have not covered yet. This person, the AP Bio Penguin, she is an AP test reader, and she knows what those AP readers are looking for, and it can be very, very helpful. So I highly suggest looking at that, especially once we can complete Unit 5, and especially right before you take the test, a little cram session. That would be very useful. Crash Course is always fun to watch. It discusses heredity in an accessible way, but it's somewhat oversimplified. But it's useful to gain a basic understanding of general heredity. It won't be super useful for AP Bio, but at least if you're really, really struggling, it'll give you a more accessible way to understand it. And then you have the Bozeman Science review that reviews everything we discussed today, and it also introduces probability mathematics of single traits to determine probability of certain trait combinations. And here are my citations for this video, and I appreciate you listening. Thank you very much.